Greeting friends, we have finished the reactions of glycolysis and gluconeogenesis. So let's move now to the coordinated and reciprocal regulation of glycolysis and gluconeogenesis. So we start with the question, why regulating glycolysis? Why there is a need to regulate glycolysis? So to answer, we can say rate of conversion of glucose to pyruvate is regulated to meet two major cellular needs. First is production of ATP, and the second is provision of building blocks for synthetic reaction. And if there is any problem in regulation in glycolysis, these two can be disturbed. There can be problem in the synthesis of ATP, and the building blocks for the synthetic reactions cannot be produced, because we are producing the intermediates in glycolysis, and those intermediates are used for the production of, or the synthesis of, or the reductive synthesis, I must say, for other processes like NADPH is used in pentose phosphate pathway, and acetyl CoA we use in citric acid cycle, also in the fatty acid synthesis. So next is the key points in regulation of glycolysis. We start with, in metabolic pathways, enzymes which catalyze the irreversible reaction are potential sites of control. So we have to focus on those enzymes which are catalyzing the irreversible reactions. And in glycolysis, we know what are those three enzymes. The first one, hexokinase, that catalyze the first step of glycolysis, conversion of glucose to glucose 6-phosphate. The second one is phosphofructokinase, that is the PFK, catalyzing fructose 6-phosphate to fructose 1,6-bisphosphate. The third one is pyruvate kinase, that converts PEP to pyruvate. And hence we can say these enzymes would be expected to have regulatory and catalytic role. Now there are types of regulations we are regulating here. So different types of regulation you can see. The first one would be allosteric regulation, that occurs in milliseconds. The second is covalent modification. This is something in which phosphorylation and dephosphorylation would be there, and it occurs in seconds. And the third one is transcriptional control. It occurs in hours. We would not discuss this in this video because this is beyond the scope of this lecture. This is about the, we can say, molecular biology. I just tell you what is transcriptional control. Here is simply there is an increase or decrease in the frequency or the number of enzyme molecules through their synthesis or breakdown, through using the central dogma of biology, that is DNA to RNA and to protein. Okay, so the next here is PFK. We start with phosphofructokinase, which is a key enzyme in control of glycolysis. Why? Because it is the most slowest step, rate limiting and committed step. You have to focus on this committed step. Why? Because when we say it is committed, it simply means if it has converted fructose 6-phosphate into fructose 1,6-bisphosphate, this product is not going to form the fructose 6-phosphate again. That is simply committed. Next is, we can say, now we are going to discuss the effects of certain molecules on this enzyme. The first one here is allosteric effect of ATP and AMP. First we start with ATP, that is, if there is a high level of ATP, then enzyme would be inhibited, and result would be the affinity of the enzyme for fructose 6-phosphate would be lowered. Hence the reaction would not occur, that is fructose 6-phosphate would not be converted into fructose 1,6 bisphosphate. And we can say more here, that is hyperbolic binding curve into sigmoidal. See here, this is a picture. You can see here there is a green color line and red color line. This green color line demonstrates the low ATP. When there is a low ATP, the curve is here hyperbolic. Okay, that is the reaction velocity is high. And on the other hand, if it is red, that is high ATP, then there is a deactivation of this enzyme, which we have just seen. So this is just for the understanding. Let's move to the next slide. Now the question is how it happens, how this ATPE or AMP is increasing and ATP is decreasing. We can say PFK has two regulatory sites. PFK, which is the enzyme, is having two regulatory sites with different affinities for ATP. Now ATP is acting as both as substrate and inhibitor. So when ATP binds to a specific regulatory site, okay, which is distinct from the catalytic site, it inhibits this enzyme simply. Now the effect of AMP. The effect of AMP is in opposite. It is acting in the opposite direction. That is, reverses the inhibitory effect of ATP. Activity of the enzyme increases when the ATP to AMP ratio is lowered. When AMP is more and ATP is less, simply means its ratio is lower. And what more we can say about this is that glycolysis is stimulated as the energy charge falls. So it simply means when there would be more AMP, then the charge would be less. Why? Because ATP is replaced by AMP, and the result is that the phosphate groups have been lost. So charge is less. So glycolysis is stimulated. The third here is the effect of pH. When there is a fall in pH, it inhibits PFK. And this inhibition by H positive, if there is a less pH, it means there would be more H positive ions. So these H positive ions prevents excessive formation of lactic acid. Lactic acid that is formed when there is anaerobic glycolysis. And the result is that when H positive prevents excessive formation lactic acid, the inhibition of this lactic acid formation would protect the muscle from damage, which would have been caused by the accumulation of, we can say, this lactic acid in muscles. And also we can say acidosis, that is drop in blood pH. So the physiological pH of blood would be maintained. There is one more point here, that is glycolysis furnishes carbon skeleton for biosynthesis. Biosynthesis of what? Other molecules, like we just have discussed about the pentose phosphate pathway and the acetyl-CoA. So these building blocks are abundant or scarce, so there would be a signal for that. So this would regulate glycolysis also. We can see through an example. So a high level of citrate, if there is high level of citrate, it simply means we do not need citrate, so we would stop glycolysis, a signal to stop. That is, the precursors are abundant, no glycolysis, हमें नहीं जरूरत glycolysis के, रोक दो. Now let's move to gluconeogenesis, the corresponding step in gluconeogenesis. We know fructose 6-phosphate converts itself into fructose 1,6 bisphosphate. Now we are converting this fructose 1,6 bisphosphate to fructose 6-phosphate back by bypassing, and the enzyme here is fructose 1,6 bisphosphatase. Okay, this is inhibited by high AMP. So this is opposite of glycolysis. AMP, high AMP stimulates glycolysis. It inhibits gluconeogenesis. Thus we can say these opposing steps in glycolysis and gluconeogenesis, those catalyzed by PFK and FBPase1, okay, are regulated in a coordinated and reciprocal manner. So this is a picture here. You can see the molecules which we just have studied, ATP, ADP, AMP and citrate. Their presence or their absence would affect gluconeogenesis and glycolysis differently. Now there is a very important molecule which we haven't seen yet. It is fructose 2,6 bisphosphate. Okay, this is potent allosteric regulator of PFK1 and FBPase1. I must tell you that please emphasize on the numbering of these enzymes, because you are going to see the other variants of these enzymes too. Let's know something more. Hormonal regulation now comes into play. So far we have only seen these enzymes. No hormone we have seen yet. So for that we must know what is the role of liver. It maintains constant blood glucose level. How? By additional regulatory mechanisms which we would be studying now. For what? To coordinate glucose production and consumption. Okay. How? We discussed two scenarios now. When there is a low blood glucose, what would happen? Glucagon hormone would be released and it would signal liver, hey, रुक जा भाई, अभी ग्लूकोज जो है ना उसको तू अपने लिए इस्तेमाल मत कर. Don't use glucose for yourself. Stop it, man. We need more glucose, so produce more. Okay, so how this is achieved? Through glycogen, which is stored in liver. That is glycogenolysis. There would be breaking of this glycogen to produce glucose, and through also gluconeogenesis. The second scenario here is when the blood glucose level is high. So this would be the opposite. Now the hormone would here be insulin. It would signal liver, अरे भाई वापस आ जा, सॉरी यार वापस आ जा, हमें रिवर्स करना पड़ेगा. Use glucose now. You can use glucose. Okay, and as a fuel or precursors, you can use glucose for yourself also, and you can make precursors from this glucose too. This rapid hormonal regulating of these processes is mediated by fructose 2,6 bisphosphate, which is allosteric effector for enzymes PFK1 and FBPase1. Now this molecule is going to regulate these two enzymes which we have studied. But how? Let's see. Fructose 2,6 bisphosphate binds to PFK1, and when it binds, it increases enzyme's affinity. For what? For its substrate. And on the other hand, it reduces affinity for its inhibitor ATP and citrate. If it is absent, what would happen? PFK1 would be inactive, and on the other hand FBPase1 would be active. Result would be gluconeogenesis would be favored and glycolysis would be stopped. The second scenario would be vice versa, opposite of that. If it is present, PFK1 would be active and FBPase1 would be inactive. Result would be opposite of what we have studied. That is, gluconeogenesis would be disfavored and glycolysis would be continued. What is more here? Now the concentration of fructose 2,6 bisphosphate by its formation and breakdown. You know what we are doing? We are going into deep background slowly. हम धीरे धीरे जो चीज़ें हम देख रहे हैं उन चीज़ों को रेगुलेट करते जा रहे हैं. पहले एंजाइम्स को रेगुलेट किया, फिर जो उनको रेगुलेट कर रहा था फिर उसको रेगुलेट कर रहे हैं. दैट इज फ्रक्टोज 2,6 बिसफॉस्फेट नाउ. हाउ? बाय इट्स फॉर्मेशन एंड ब्रेकडाउन. Its formation and breakdown is done through regulation by phosphorylation and dephosphorylation, that is covalent modification by PFK2 and FBPS2. So these enzymes which we have seen already, but the difference here is of number 2. Formation by phosphorylation of fructose 6-phosphate by PFK2. Phosphorylation is occurring at this second carbon instead of first. Breakdown would be dephosphorylation of this fructose 2,6 bisphosphate phosphate by FBPS2. So we can say if there is phosphorylation, there would be presence of fructose 2,6 bisphosphate. So it means if it is present, glycolysis would be continued and gluconeogenesis would be stopped. And on the other hand, if there is dephosphorylation by the enzyme FBPS2, there would be absence of F2,6 bisphosphate. So there would be no glycolysis and gluconeogenesis would be favored. It would be stimulated. So the next here is, so there is a flow chart which demonstrates the presence and absence of this molecule fructose 2,6 bisphosphate. You can see that. And the summary we can say, yeah, this is important. I forgot to tell you that these enzymes PFK2 and FBPS2, these are present on the same enzyme. So this is one enzyme which have two functions, which is the dephosphorylation and phosphorylation. Now we are going into the deepness of this pathway. This is started from CAMP. You can see here, this is our CAMP here. Increase in CAMP signals glucagon to be produced. When it is produced, it signals this enzyme, CAMP dependent protein kinase, and this enzyme is going to phosphorylate because it is kinase, so it would phosphorylate this bifunctional enzyme which is containing this PFK2 and FBPS2. So this would be causing inactivation of PFK2 and activation of FBPS2. If PFK2 is inactive, glycolysis would be discontinued and gluconeogenesis would be favored. You can see here because there is a decrease in fructose 2,6 bisphosphate now because FBPS2 is active, this in green color here. This would, you can say, dephosphorylate, would remove the phosphate group and this glycolysis would be disconnected, the link would be disconnected. And on the other hand, if there is a dephosphorylation of this particular enzyme which is containing two enzymes here by phosphoprotein phosphatase here, when insulin is acting on this enzyme, it would be the opposite here. You can see PFK2 is active now and FBPS2 is inactive. Here this is removed, phosphate group is removed, and the result would be opposite, that is glycolysis would be continued and gluconeogenesis would be disfavored because there is an increase in fructose 2,6 bisphosphate. So this was all about the regulation of PFK. In the next video, the regulatory part of the regulation would be of, we can say, the other enzymes, that is hexokinase and pyruvate kinase. Thank you.